Perhaps the most salient characteristic of skeletal muscle is that a skeletal muscle cell is also a skeletal muscle fiber. So skeletal muscle cells are very, very long cylindrical structures that, for the most part, extend the length of the muscle. As we're going to see as we look at some histological images in just a moment, they are specialized, of course, for contraction. These long cylindrical cells would not work very well if they only had one nucleus over such a large volume of cytoplasm. You would think the axons would pick up on that trick, but no. So the skeletal muscle cells are multinucleate. And the nuclei tend to be peripherally positioned along the margin of the cell. Another important difference between skeletal muscle and the other two classes of muscle up here is that skeletal muscle is exclusively neurogenic. So we've talked about this before. By that we mean that skeletal muscle cannot contract in the absence of a neural signal. It depends upon a neural signal to contract. The other two types of muscle, to one degree or another, have the gap junctions. What is a gap junction? Right, it's a direct cytoplasmic bridge between adjacent cells. So we don't really have that in skeletal muscle. The skeletal muscle contractions tend to be more powerful than the contractions of the other two classes of muscle, but they are also very short-lived, and they tend to require a lot of energy, generally speaking. For that reason, skeletal muscle is much more prone to fatigue. And that fatigue can occur even in the absence of adequate fuel. And of course, anatomically, skeletal muscle is present for the purpose of articulating the skeleton, moving the joints, and a few other places as well. For example, initiating swallowing and some other areas as well. Now we'll talk about cardiac muscle in more detail later as we look at the cardiovascular system. So we'll defer a consideration there. But I would encourage you to spend some time looking over the general characteristics of the cardiac muscle, simply for comparison. And then a little later we'll talk about smooth muscle. But smooth muscle is something of a hybrid between the two in some respects. So typically skeletal muscle accounts for about 40% of total body mass because of the water component. It's a fairly dense material, over 600 muscles. Also, not only does it contribute to articulation of the joints, skeletal muscle also contributes to thermoregulation. Indeed, most of your body heat is generated by the activity of skeletal muscle. We talked about the histology earlier, and you can see the peripheral nuclei here, these long cylindrical cells that tend to be striated. Striated just means that they have bands, and actually you can see those bands at a very low magnification. You can just take a magnifying glass and walk up to your favorite steak, and in most cases you can see the striations there. But that's all that is meant by that term, the banding, and we'll talk more about the significance of the banding a little later. This is an actual micrograph, again, peripheral nuclei, striations along or at right angles to the length of the muscle. We indicated that the muscle cell is also the muscle fiber, and that because these muscle cells are so long and do have to have multiple nuclei, we sort of artificially suggest that the region or volume of cytoplasm regulated by a single nucleus is called a skeletal muscle DNA unit. So a DNA unit is the volume of cytoplasm regulated by a single nucleus. And this electron micrograph image over here sort of appears to be fake, but that's an actual image, and you can see the extraordinarily regular and precise arrangement of the various filaments and fibers we'll talk about in just a moment. You can see some other structures too. You can see the nucleus. You can see mitochondria. This is extremely, the skeletal muscle is extremely consumptive of energy, so there's lots of mitochondria packed in there. And then, of course, this infrastructure is the basis for the striations that we'll be looking at in more detail. We have a couple of illustrations here showing the organization, the hierarchical organization of the terms that we use to describe the elements and components of skeletal muscle. So for example, at the bottom down here, you can see the general hierarchy. You can pick any muscle, let's say a biceps, a brachii muscle. It in turn is going to be made up of individual fascicles. And fascicles are just basically connective tissue delimited subunits of the muscle. Each fascicle in turn is made up of many cells or fibers. The fibers consist of many thousands of fibrils, also called myofibrils. And then the fibrils are made up of filaments, and we're not even at the end of the chain there. We have to talk about the proteins that make up the filaments. So be familiar with that hierarchy. And of course, in this list at the bottom left here, I've also provided you with the connective tissue capsules that surround each of these major structures. Remember, we encountered something like this when we talked about nerves. An entire muscle is surrounded by epimysium. Remember, that is a type of deep osseal membrane that is usually just under the subcutaneous layer of the skin. The fascicle is encapsulated by perimysium, and then each cell has its own little endomysium, very analogous to the organization of the nerve. And you see something of that organization up here in the upper left. You can see, for example, in this muscle, there are individual fascicles. There would be a fascicle. Each of those is made up of individual muscle cells. The cells are the little, I guess in this illustration, sort of brown or orangish structures. And then we begin to sort of lose resolution, but you can see the tissue colored blue here would represent the connective tissue. So this would be the epimysium out here, the perimysium. And then the light blue that you see inside the fascicle would be the endomysium. And all three layers would have different levels of neurovasculature running through them to supply the structures on the inside, the structures inside the connective tissue. We'll talk more about the banding in just a moment, but here's a single muscle cell. It's being made up of individual fibrils. So the only thing I would have you pay attention to now is notice that the striation and banding that we've been talking about that's characteristic of skeletal muscle appears at the level of the fibril or myofibril. So you can see the little lines there that are present in the myofibril. Do you remember what the neurolemma is? What is the neurolemma? Well, the neurolemma site is a Schwann cell, but the neurolemma is just the boundary, sort of usually the boundary of the axoplasm. Likewise, we have a sarcolemma. In fact, we have our own specialized language to describe these muscle tissues. The sarcolemma, for our purposes, is just the plasma membrane of the muscle cell, along with in fact, with what sometimes is called a glycocalyx. Remember our discussion of the glycocalyx. So we'll just say that it's sort of an outer coat of glycocalyx, and there's some collagen thrown in there as well. But all of that forms the sarcolemma. And in these illustrations that we're looking at in this slide, this thing out here, this sort of reddish or orange thing would be the sarcolemma. Over here is gray colored. And what you'll notice is that sarcolemma is perforated. By that I mean there are little apertures or openings in the wall of the sarcolemma. And these apertures, such as you see here, you see them up here, these open into an interior plumbing system, a plumbing system that reticulates and branches throughout the entire interior of the cell. And here it is right there, at least part of it, called the transverse tubule system. So there's a transverse tubule there. In this illustration, the transverse tubules are yellow. So be sure you recognize that the transverse tubules open onto the perforations or the openings of the sarcolemma. Now, again, this whole thing would be a fiber or a cell. The whole thing is a fiber or cell here. We know that the fiber or cell is made up of individual myofibrils, which we see here. And you can see the myofibrils over here, these little pink and blue things. Right? So in both illustrations, and fortuitously for us, we can see that in both illustrations they're blue. You can see this sort of encasement that wraps around the myofibril. All these myofibrils are encased by this blue thing, which is another little plumbing system. The blue encasements of the myofibrils are called sarcoplasmic reticulum. Sarco implies muscle or flesh, sort of the Greek origin there. We've used the word reticulum before, as we talked about, for example, the endoplasmic reticulum. In fact, the sarcoplasmic reticulum is just a modified specialization of the ER in muscle cells, but reticulum implies a network of branching tubules in this case. So pay careful attention to the relationship of the transverse tubules. What does transverse mean? You have a transverse mounted engine in your Ferrari. That's what I have. Yeah, perpendicular too. So what the T tubules or transverse tubules are perpendicular to the plane of the muscle cell. The direction or orthogony of the muscle cell. And then notice that the transverse tubules are tightly, closely apposed, APPOSED, they're tightly adjoined to the blue sarcoplasmic reticulum, so that there's a very close and intimate physical relationship between the two. That's probably not the best choice of words, but I'm a scientist. I can say things like that with complete impunity, without anyone casting aspersions upon my character. So make sure you pay attention, and I think it's fairly clear in both cases. And of course, these arrows sort of tell you what's going to happen here. By the way, if this is a cell, of course, this cell is going to be embedded in some larger tissue. And you can see this is intended to be a neuron here. Remember, skeletal muscle is neurogenic. It requires an action potential coming from a synaptic terminal to cause excitation of the skeletal muscle, and that's what the blue arrows are representing. So there's going to be propagation of the action potential along the transverse tubules. But those transverse tubules are tubules, and what are they filled with? What would you put in there? Think about where the muscle is. What's inside the transverse tubules that facilitate this process? Well, there are, but we'll get to those later. The receptors are going to be very, very important. But the entire volume of the tubule, what's going to be in there? Is it going to be empty? For example, in the medieval period, it was thought that your arteries were filled with air. We think that may not be true these days. What would be in there? Well, no, that's what I was, so the cytoplasm is going to be inside. That's what I wanted to make sure you understand. The cytoplasm is sort of going to be in here. So is that clear? So this is a plumbing system that is connected to the outside world. And what's in the outside world? I can't say ours today. World. What's out there? ECF, yeah. I just want you to make sure that this is a direct introduction of the external environment into the innards, that's a technical term, innards of that skeletal muscle fiber. The cytoplasm would again be surrounding that tubule. So you really got two specialized, somewhat isolating plumbing systems that are working in a very cooperative fashion to regulate contraction of this entire tissue. But let's not get too far ahead of ourselves. Let's make sure we understand all these terms. And make sure sarcolemma, sarcoplasmic reticulum. We've indicated that there are invaginations in the sarcolemma to allow for communication with the transverse tubules or T-tubules. And then you sometimes see this word. I don't like it because it's inherently confusing to most of us. The sarcoplasm is sometimes described as the cytoplasm of the muscle cell. But what do we say is the sarcoplasmic reticulum? What is it? Yeah, and in this case, so I want to make sure you understand that there is not sarcoplasm inside the SR. There is not sarcoplasm in the SR, but most of us would make that assumption based upon the way we're using these terms. I think some of you have already told me a moment ago what is inside that SR. At least what's important. Yeah, it's going to be a repository of calcium ions and a few other things. So spend some time with these illustrations or those you find elsewhere and be sure you can sort out the physical relationship between each of these components. By the way, all of these illustrations have deficiencies of one type or another, but now we're looking at the SR and the relationship of the T-tubule to the SR at somewhat higher magnification. So the yellow here, I realize this is not technically accurate, but this would be the sarcolemma, and then that sarcolemma opens into a transverse tubule that you see here. And remember in the previous illustrations, we saw the very tight relationship between the transverse tubules and the SR. So here it is. And that's sometimes called a triad. What you're looking at there in the triad are two terminal cisternae. So here's our transverse tubule here. And then on either side of it are the cisternae. And the cisternae are just specialized regions of the SR that adjoin the T-tubule. They're having a drought in other parts of the country, so they, because of the drought, and folks living, well, this is true in the American Southwest in general, they will go to great pains to collect rainwater. When they collect the rainwater, they often do so in cisterns. So the term cistern, C-I-S-T-E-R-N, simply implies you're collecting, you're aggregating, it's a repository or a vessel for some type of fluid. But in any case, when we talk about the skeletal muscle triad, we're talking about two terminal cisternae, both components of the SR, separated by a transverse tubule. Of course, you have to imagine this in 3 dimensions. I reorganize these things. So what's going to happen essentially is that there's going to be some type of neuromuscular junction here. That's going to cause an action potential to be propagated away from that neuromuscular junction into the T-tubule. And then by virtue of this very close relationship between the T-tubule and the cisterns of the SR, the action potential entering the T-tubule is going to excite some type of change in the SR. So that's really as far as we've gotten in our story. We have the anatomy to work with. And then back in the back there, you see our principal actors. You see the filaments back there. We're going to talk about the thin and the thick filaments. And so the purple thing back there is going to be a thick filament, otherwise called myosin. And then the red things are going to be the thin filaments, otherwise known as actin. Yeah.
So the action potential
Well, it doesn't, it's traveling along the membrane, but you can imagine, again, we talked about capacitance and so on, we talked about action potentials in neurons and so on, remember that, subsequent conduction. This wouldn't work if we just had one little phospholipid bilayer going down here. So the action potential is being propagated along the membrane, but it requires this entire structure for that propagation to occur. So this puts together, this image sort of puts together some of the things we've been talking about in their respective hierarchy. Hopefully at this point you can see the relationship between the muscle, the fascicles, the connective tissue and casements, and of course the extensive neurovasculature that's required to keep all this working. And then when we get down to the level of the myofibril, which is here sort of a green and gray colored structure, we begin to see how that banding arises. And we're looking at the relationship of the green and the purple or whatever color that is down here. In this illustration, the green, of course, is the myosin filament or the thick filament. And the purple are the actin filaments. And so it's the relative arrangement of these structures that's responsible for the fibrillar striations that show up in the skeletal muscle. We have several illustrations of this, but it really doesn't matter. They're about this whole thing here, from this point over here to this point over here, is about 2 microns in length. And that whole thing then is called a sarcomere. Sarcomere. So we have a lot of words beginning with sarco at this point. The sarcomere is bounded by z-discs. So between each pair of adjacent z-discs, we have one sarcomere, at the center of which is this lengthwise arranged myosin filament. Again, imagine this in 3 dimensions, and in this myosin filament, you'll note that at the terminate of each end of the myosin filament, there are these little golf club looking extensions. These are called the myosin cross bridge heads. And you'll notice the myosin cross bridge heads appear to be attached to the actin filament. What will happen, of course, in the course of muscle contraction is that these myosin cross-bridge heads are going to change conformation. It's called the walk-along theory. They're going to attach, change conformation, detach, reattach, and change conformation. It just keeps going back and forth. And as that happens on both ends, the z-discs come closer together. And of course, as each individual sarcomere contracts, the entire fibril, and in fact, the entire muscle contracts. So we've talked about the 2 filaments that make up the skeletal muscle, and this should look familiar to you. Do you remember what we called these types of things? Well, kinesin and dynin were both types of, what were they? They were an example of a motor protein. So we're looking at here a fundamentally very similar sort of thing. But in this case, this is the myosin cross-bridge head we're looking at here. This is the myosin cross-bridge head of the myosin filament. And it has the ability, as you can see here, to walk along the actin filament in much the manner, that very laborious, cumbersome manner that we saw with the dynins and the kinesins. And an ATP, some other things we'll talk about, dependent fashion, to bring the 2 Z lines closer together. Now, the myosin is relatively straightforward in its organization. It's this, by the way, make sure that you understand the myosin filament is made up of individual polymeric myosin proteins or myosin molecules. So the myosin molecule itself is a polymer. Looks like, again, you can sort of see here, it looks like a golf club. The filament, and this gets if anything, only more complicated when we start talking about actin. The filament then is also called the myosin filament, but it represents an aggregation in 3 dimensions of many hundreds of these myosin proteins. Later, not so much in our discussion of skeletal muscle, we'll talk about heavy chains and light chains. But notice the heavy chains contribute to the shaft, the light chains of the myosin molecule contribute to this little inflection down here. We say that myosin has ATPase activity, and by that we simply mean that when ATP attaches to myosin, the myosin has the ability to hydrolyze the ATP, break it apart, ATP inorganic phosphate, and in so doing, use that power, use the energy extracted from the phosphate to change its conformation, to change its shape. So the ability of the myosin molecule to change shape is ATP dependent, although as we talk about muscle contraction in more detail in just a moment, we have to be careful with our wording, and we'll return to that. All right, so that's the, just again, make sure you understand the distinction between the myosin protein and the myosin filament and the relationship between the two. All right, let's look at the actin. And you have an illustration here. Here, we'll look at several illustrations showing the same thing. Once again, we have the same problem. We have to differentiate this rather complex polymer, this rather complex filament, from its individual components. So this whole thing then is the thin filament, and it's made up of several important constituents. First of all, the actin filament is made up of polymer actin proteins. Each one of these red spheres is called a G-actin or globular actin. The G-actins are sort of strung together in that helical sort of fashion there to form F-actin. So when you string them all together, they become filamentous actin or F-actin. And then, kind of like you can see a sort of a double braided wire that's wrapped around again in a helical fashion, the F-actin. That is tropomyosin. That's a long linear protein as well. Tropomyosin, which in this illustration appears to be gray or silver. And then finally, attached at various points along the tropomyosin and the actin filament, are the troponin proteins. Troponin. And they've been playing around over the past 5 or 6 years with the designations of these protein. The troponin. There's troponin C, troponin I, and troponin T. And those are sort of explained here. The troponin C is going to be important in our discussion because that's the species of troponin that binds calcium. Calcium, as we're going to find out, is essential for binding between the myosin cross-bridge head and the actin filament. Troponin I, sometimes used to be called tropomyosin troponin, acts to inhibit the binding of the myosin. So it basically is a competitive inhibitor and blocks the binding of the myosin cross-bridge head to the actin. And then troponin T, medical students sometimes remember T for tether, joins the troponin to the tropomyosin. Yeah.
Are these troponins the same kinds that are released when you have ischemia in the heart?
So you're talking about a blood test, a blood assay for troponin. And why would it show up in a cardiac infarction, would you say? Well, what is causing, where was that troponin to begin with?
Cardiac tissue?
Yeah, it was in the cardiac tissue. And of course, now the reason it's in your it's circulating at higher concentrations in the blood is because what's happened? Yeah, yeah. So yeah, this can happen too in skeletal muscle. Troponin, there are many different types of troponin. We're dramatically simplifying this. But this can happen too, especially in farmers. Extensive bruising to the muscle, but it's not generally diagnostic for that purpose. You generally, when a muscle has been traumatized, you generally know that. But in the principle is the same. It's fundamentally the same type of troponin. We're going to see, though, that troponin has a slightly different role in skeletal muscle. All right. So we pretty much sketched out our black battle plan here. The idea is to get these two Z discs of each sarcomere to come closer together, and in so doing, that will make the fibril shorter. That is, when we make the fibril shorter, we're making the entire muscle shorter. And we said that the way that happens is when these individual myosin cross-bridge heads initially attach to the actin filament, change conformation, detach, and they'll go back to the original conformation, and they'll just complete that process over and over again. Again, that's called the walk-along theory. And as that's happening again in 3 dimensions, this Z disc is brought closer to this Z disc. So we need to work out now how it's possible for these myosin cross-bridge heads to achieve that. By the way, before we do that, you'll notice you need to be somewhat familiar with the terms that are used to describe the striations of the fibrils. So we've talked about the Z disc, all right? So the Z disc would be this point here and this point here. In this illustration, in this particular illustration, notice that the actin filaments radiate in opposite directions from each Z disc. So here's our Z disc or Z line. And then those little red lines that you see there, those are the actin filaments. At the center of the sarcomere is the M line. A lot of these abbreviations come from the German, as I recall. So M was beta. I think Z was Vita. Between or something. And so so we have one sarcomere. So what I want you to pay attention to are these remaining designations up here: the A band, the I band, and the H zone. You can look at that illustration or either one of these. Neither one of these are particularly great. I'm going to use this one. So the blue or the purple here represents what? What are the blue? What are the purple things there? Those are the myosin filaments, right? And at the center of the myosin filament, the array of myosin filaments is the M line. So how would you characterize this region called the H zone? How would you describe it in terms of its position and relationship in the sarcomere? Anybody want to give that a try?
The distance between the myosin filaments that come from opposite
You say the distance between the myosin. In that H zone, do you have any actin filaments? No. So you only have myosin filaments. So we could begin with, it's that portion of the myosin filaments that what? That does not or do not overlap with the actin filaments. All right, so there's no overlapping of the myosin with the actin in the H zone. What about the I band? How would you characterize that? All right, good. The I band is a region of actin filaments. only. There are no myosin. There's no myosin in there. And what else could we say to differentiate it from the other regions? This is a region of actin that does not overlap with the myosin. All right, so make sure you see the distinction between the I band and the H zone. And we left the easiest one for the end. What's the A band? Yeah, that's just the length or the length of the myosin in the sarcomere. Corresponds exactly to the length of the myosin. Now remember, during muscle contraction, the Z discs are going to come closer together, and of course in muscle relaxation they're going to separate. If that's true, between the A band, the I band, and the H zone, which of those will change length during muscle contraction? Will they all change length? Will the H zone change length? Yes. Will the I band change length? Yes. Will the A band change length?
No.
No. All right, so make sure you pay attention to that. Make sure you realize that. The A band does not change length because the filaments themselves do not change length in the course of myofibril or muscle contraction. The filaments do not change length. Neither the thick filament nor the thin filament change length. Rather, the degree of overlap, the distance of overlap between them, changes length during contraction.
So which one changes?
Um,
The H on shorten?
Yeah, the H and the I change length. The A band does not. H and I do, A does not. And you see that written there. Just want to make sure you see how we derive that. So really, again, the bottom line here is that what is happening during sarcomeric contraction is the region of overlap between the thick and thin filaments changes. And of course, you can see this is just this is about, I don't know, about 25 power. It's very low power. You can see the striation. We can actually break down the Z lines and the I bands in this muscle, skeletal muscle tissue.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
And that's kind of an important point because the degree of overlap really determines the efficiency, among other things. It determines the efficiency of skeletal muscle contraction. If you look at these graphs here, this is a graph of tension over time. Another way to say that is this is a graph of force, the force of contraction over time. So you'll note in this particular preparation here, you can see the muscle length, they get a force, a dynametric force of about 10 units, whatever those units are, let's say pounds. It's kind of excessive. Here's this one, this particular preparation provides a contractile force of 20 pounds, and then over here we're back to 10 again. So I guess my question for you, as you look at the explanation of the graph at the top there, why is this one so much more efficient than the other two? What is indicated by the representation of the sarcomeres here that would provide us some clue as to why muscle length 2 is to be preferred if we want to optimize our force of contraction? And you keep in mind this if you ever ran track or whatever, right? When you began to do the sprint, you didn't start like this, right? You were in the blocks. I felt better after that. You're in the blocks and muscles are partially contracted. if a football player at the line is lining up, although they're changing now, they do stand up at the line now. They're crazy. But back in my day, they would get down in a crouch position. Same with wrestlers. Why?
There you go.
Explain. Not so much we're talking about the filaments here. So what's the problem here? Yeah, you've contracted it, and you can't contract it anymore at this point because the Z lines are so close together. What's the problem over here? Yes, there's no overlap, or there's insufficient overlap between the myosin cross-bridge heads. The muscle is too stretched. You know, you can imagine if you had to, of course, there are other kinesiological reasons for this, but if you have to lift a weight, do a curl of weight, is it easier at this position or this position? It's much easier in the middle, right? In part because of the optimal degree of overlap. Here we have all of these myosin cross-bridge heads. They're ready to go. There's plenty of room for contraction, and each one of them, every one of them, is attached to the actin filament. Is that clear that how we get the? And if you actually, I realize this is going too rapidly, but you see a couple of things here. You can see the I band and the H zone changing length with contraction. And if we were to slow this down, which I didn't do, if we were to slow this down, we would see that the optimal force of contraction down here in the lower right is in an intermediate point, a point when it's not fully stretched and not fully contracted, but somewhere in the middle. And Flash always crashes my computer, so go to the next one here. All right. So there's a somewhat arbitrary distinction between what are called structural proteins and regulatory proteins. We generally refer to regulatory proteins as those that are actually involved in the process of muscle contraction. But there are also some structural proteins that contribute to this, and these are going to be important for you as a PA because a lot of diseases, a lot of muscle pathologies, for example, depend upon these proteins being prevalent and available in the skeletal muscle tissue at appropriate levels. Titin, for example, some of these are more important than others. If you look at in this animation on the lower left, you see those little yellow things that look like springs. If you were to, I don't know, interpret the function of those yellow springs to a fifth grader, what would you say they're doing? Fourth grade. First grade. Why are they there? Let's take them away. And by the way, you'll notice this yellow thing actually goes all the way from one side of the Z line to the other. All the way. Yeah. Now, Trenton is saying that it's sort of acting. Usually, the comparison is to a spring. I hope you realize this is a molecule. It's not. We're not talking about macrophysiological phenomena here. But the idea is that titin is helping to optimize, and as we saw in that previous illustration, help to keep each individual sarcomere at just the appropriate length. It's preventing it from stretching too far at the molecular level. You have other things to do that perform the same function at the tissue level, and also helping to prevent it from contracting too far as well. So that's the general function of titin. The nebulin, this is one of these things where 10 years ago there was a very precise definition of what nebulin did in the textbooks, and of course the more we learn, the less we know. Now, the nebulin, I don't know if you can see it here, it's that sort of turquoise line. About all that you'll read now is the nebulin is thought to contribute to the stability of the actin. And what we do know, if it's not present, the muscle doesn't work properly. mean, you've heard of muscular dystrophy. There are many types of muscular dystrophy. This dystrophin protein is a product of one of the, I guess it is the largest gene in your genome. And this too, we are not entirely sure of how it functions, but it seems to play a very important role in anchoring or maintaining the relative position of each sarcomere to an adjacent sarcomere. Keep in mind that the striations that you see in skeletal muscle are at the fibrillar level, but you're looking at that muscle at the cellular level, at the fiber level. So what you can infer from that is that the individual fibrils must be precisely lined up, very precisely. It seems to be an important contribution of the dystrophin. All right, we'll take a break there.